Thank you for the invitation. As Gabriele requested, I will start with a broad overview of super resolution microscopy. So I'll give my historical perspective of the field and some hints to what is ahead, I hope. Before we start off, like I said, it's going to be an overview, so very little of my work. But in the end, I will show a few of the things we have been doing over the last few years. And since some of the work led to IP and spin-off companies, I just mentioned this just to be very clear and open about this. Okay, I think it's fair to say that the main way we interact with the external world is through vision. It's fair to say that our eyes are very sensitive and good detectors of photons, and that our brain is wired very impressively to process images. That will be a talk in its own right. But like any optical system, our eyes, as good as they are, they have limitations. And the main limitation is the resolving power of our eye, which boils down to something like 100 micrometer or just below, which is about the thickness of an hair. So many of the aspects in the microscopic world that we see find their origin in features that are much smaller than the resolving power of our eyes. For example, this green color in the butterfly is owing to a nanophotonic organization of pigments. The malfunctioning of the airplane can be due to a microscopic crack in the metal of which the airplane is built up. And the fact that we get sick can be due to viruses or aerosols containing viruses, as we all know very well now, that we simply cannot see by our eye. And that's why people have invented microscopes, just to magnify details in images that are not visible to our naked eye. Microscopes have come a long way. First reports date back to the late 1600s. Very often they're linked to work of Leeuwenhoek, a Dutch trader actually, who invented a microscope for inspection of cotton fibers in cloths that he was selling. And over the years, they have grown to instruments that we come to know very well. Now, if you use in your microscope fluorescence as your contrast mechanism, you're doing fluorescence microscopy. And I think it's fair to say that fluorescence microscopy has become tremendously popular in the life sciences and more also in material sciences. And you all have seen images as the ones depicted over here that are not only very beautiful and colorful, but also contain a lot of information. And there's many good reasons why fluorescence microscopy has become so popular. It's selective, non-invasive, easy, although that is a relative notion, of course. It allows you to look at living samples. It has a huge dynamic range from experiments that go on the nanosecond time scale all the way to experiments when you set them up correctly that can last days and weeks. And it's a very sensitive technique. I think it's the only technique that is single molecule sensitive on a routine basis. But again, like any optical system, also fluorescence microscopy has its limitation, and that is a still limited spatial resolution. And obviously that is a problem, especially if you look at the life sciences, where the key building block of cells, for example, are proteins. And proteins are typically in the range of a few nanometer, let's say 5 nanometer. The resolution in your fluorescence microscopy is about 200 nanometers, so you still 2 orders of magnitude off. And we all know, of course, that the reason for this limited resolution is the phenomenon of diffraction. Now, over the last 15 years or so, the last 2 decades, people have found ways to overcome this physical law of diffraction limit, or maybe it's better to say circumvent this physical law. And as you all know, this has resulted in 2014 in the Nobel Prize for Chemistry for 3 gentlemen that you see here projected on the screen. In my view, this circumvention, or this shattering of this physical law didn't come overnight. I'm gonna make a parallel now with the field of single molecule microscopy, the field that I've been very active in since the mid-90s. Also, that field is not a field that was all of a sudden there by just one paper or just one invention. It's the legacy of many people that has led to a momentum and an idea that was out there and that was there to be realized. So if we look at the history of single molecules, I think many people will agree that these are 2 seminal papers. The Mörner paper and the Orit paper in the late 80s, early 90s. And many people see this as the birth of the field of single molecule spectroscopy. But if you look a little bit in detail, you see that there was notions of detections of single molecules already quite a few years before that. People like Dick Keller have been trying to look at single molecules in solution. And actually in 87, there was already a report where they looked at individual phycoerythrin molecules that are multichromophoric antenna systems, but nevertheless, they could detect single molecules of that. And if you look even further, you see that the famous polymer physicist Degenn already in 78 in his famous book was talking about single molecule experiments that would allow to visualize or to prove or disprove his gravitation theory. And actually it took till recently, at least for synthetic polymers, so this is a labeled synthetic polymer, to visualize what that experiment that he was suggesting back in 78. And if you really dig in deep, you see that probably the first single molecule experiment was a single enzyme experiment by Boris Rodman already back in 61. And this experiment basically was largely forgotten. But actually, I think this is the first real single molecule experiment that I have seen. And actually, I think with the field of super resolution microscopy has a parallel evolution. Of course, super resolved images in the near field regime with scanning near field microscopy were taken in the 90s already. And I think this specific configuration actually, at least in my view, that's when I got aware that it should be possible also in the far field to get super resolution. Here you see that excitation is done in the near field regime, but detection is done in the far field. And you see with this blue or purple dashed line that the diffraction limit was already overcome in this kind of schemes. And actually, Betsy, who was very active in that field in the 90s, in 95 already proposed a method where he basically said that if you can resolve individual emitters in a diffraction limited spot, you would be able to do far field super resolved imaging. Of course, there was also the paper 1 year earlier of Stefan Hell and Wichmann that pretty much said that with stimulated emission, you also should be able to overcome the diffraction limit. And afterwards, it became clear that IDs at a certain moment are just there, and very often IDs originate around the same time from different people. Turns out that already in 86, Russian scientist, Okinon, already also pointed towards super resolved images with stimulated emission depletion techniques. Now, next to these ideas that were around, I think the development of fluorophores, new probe molecules, also has been key to the development of super resolution microscopy as we see it. Organic dyes have been synthesized that are more stable. Linking chemistry has been optimized. New fluorescent proteins, genetic labels have been developed, and probes are being developed still as we speak in the form of color centers in diamonds, metal clusters, and so on and so forth. And that also was key to the development of some of the super resolution technologies that we know nowadays. So let me briefly go over what is out there now in super resolution microscopy. There's much more. You see that in the program that has been compiled for today. And once the box is open, people don't stop inventing new super resolution schemes in this case. So let me start with single molecule based technologies because that is closest to my field. Key people in there are depicted over here: Zwang, Erik, Hess, Marcus Sauer, and a little bit forgotten, but also Hochstasche, basically the inventor of paint, published work already back in 2006. So except for Marcus, who published in 2007, his direct storm, all these people published single molecule based super resolution microscopy in 2006, around the same time, which is quite remarkable, I think. And you all know how this works. If you're sure you're looking at a single molecule, you can pinpoint the position of the single molecule in the diffraction limited blob by fitting, for example, a 2D Gaussian. That works pretty good. And depending on the signal to noise ratio, you really can very accurately pinpoint the position of the emitter in that diffraction limited blob. And if you combine that with a stochastic process, and virtually all processes are stochastic at the single molecule level, you gear it up for super resolution microscopy. And I have a little animation here that I always show to my students. So what you need to do in single molecule based super resolution microscopy is in the densely labeled sample, you switch on a subset of emitters. You make sure that you always have single molecules. You apply that mathematical fitting trick, and you can reconstruct with better than diffraction limited resolution the object that you want to study. Now, was 2006 the first time when this was done? The actual answer is no. There was prior imaging methodologies that were doing actually the same thing. If you look at the paper in '94, a method was proposed that's called NALMS, nanometer localized multiple single molecules. And there is things like Fiona, Shrimp, which are all basically the same thing, single molecule based localization microscopy. We did a little contribution there as well. Also based on stochastic photobleaching, we imaged these dendritic molecules with very high accuracy. And using the bleaching approach, we could at least get an image of a projection of that molecule. But I think the problem with this prior publications is that they don't provide context, and that's of course very important. If you think back, the image of this dendrimer or the image of that piece of DNA is just a collection of dots, and that doesn't appeal and that doesn't say much to people not working in the field. So the real clever thing of Erik Betzig in 2006 in this paper on palm microscopy, in my view, was that he provided context. he provided context for this features that you show in image B, the super resolved mitochondria, basically, that he was showing. And he compared it to a proven technique that has the resolution that he was claiming, namely electron microscopy. And so that's still an important thing, I think, in super resolution microscopy based on single molecules, provide the context. And here's another example. If you don't know in image D that you're looking at receptors in nerves, if you don't provide that context, it's just a collection of dots that you're looking at. So that's a message that I want to give, I think, is that context is very important in this kind of imaging. The second super resolution modality that is widespread and well known, I think, is stimulated emission depletion microscopy. Like I said, introduced by Stefan Hell. I don't think I need to go to the principle. You use a depletion wavelength in the shape of a donut to basically engineer your point spread function at arbitrary size and to record better than diffraction limited images. Stefan has basically extended this concept of STED to what he calls resolved, where other transitions are also considered and also can be used to get super resolution. And actually, this is a picture that I always like to show. It's conference in ICP conference in Naha, Japan, 2003. And if you look carefully, you see a couple of famous scientists. You see Marcus Sauer here with his wife, Stefan Hell here at the left of the picture. You might see a younger version of myself somewhere in this picture. Turns out that in the conference dinner, by accident, I ended up next to Stefan. And so we started discussing his lecture, the first time that I had seen his lecture there on STED microscopy. And at a certain point, I asked him, is this not amendable to do FCS in smaller volumes? And he looked at me and he said, yes, that's exactly what we're working on. And indeed, 6 years later, STED FCS was reported in this Nature paper. So I always tell my students that I had at least a little contribution to the development of STED FCS. Anyway, from STED FCS, it's a small step to Stefan's newest microscopy, super resolution microscopy modality. So instead of using the undepleted zone, he started using the depletion beam to basically pinpoint the location of individual emitters in a modality that he called MINFlux, minimum photon flux super resolution microscopy, which, if you look back at it, it's a clever idea. I think it's more a tracking modality than really an imaging modality, but it can be used in both ways. And it's a clever, although technical, difficult implementation of super resolution microscopy. I'll skip this slide. So a third modality that is widespread in the meanwhile is structured illumination microscopy. I started using it only a couple of years ago. I'll come back to that later on. Invented by the late Gustafsson, who passed away, unfortunately. And basically, he came up with an idea to realize super resolution in frequency space. So this is Matt's paper in 2000. I think that's the first record of structured illumination microscopy. And like I said, you work in frequency space. So we used to images like this. If you convert that to an image in frequency space, you get this frequency histogram, basically. Now, what is resolution in frequency space? Well, that is the number of frequencies that you can get to your optical transfer function, which is basically the equivalent of your point spread function. And you see, if you have limited amount of frequencies that pass your optical transfer function, your resolution gets worse. So what structured illumination is about is that if you overlay the image you want to visualize, with a known structured pattern, you generate new frequencies in the moire pattern. It's also shown here. So if you rotate your fixed structured illumination field over your sample, you see all kinds of new frequencies. And basically what this boils down to is that you pull in new and more frequencies to your optical transfer function, and as you have seen in the image before, this results in a better resolution. And I think this is a little movie that shows or compares normal wide field image in Drosophila melanogaster cells compared to the SIM image. Now we jump forward a couple of years, and Jörg Enderlein from Göttingen, a good friend, he came up with a couple of ideas on super resolution microscopy that I think are worth mentioning. So the first implementation he did together with Shimon Weiss when he was on a sabbatical in LA is what is called SOFI, super resolution optical fluctuation imaging. And in a nutshell, what you do is basically, this is a little movie, if you have labels in your cell that allow to generate these fluctuating signals, what you can do is in each pixel of your image calculate a correlation function. Technically, it's actually a cumulant, but for a second order cumulant that is equal to a autocorrelation, and an autocorrelation is something that most people are familiar with. And it can be shown, if you do that in the right conditions, that you can get an improvement of your resolution in the case of a second order correlation with square root of 2 in your image. And since it's a correlation analysis and background is uncorrelated, you get for free a filter. You filter out basically your background, and that is shown here. This is a wide field image of a cell in which a protein TACAP was labeled with DRONPA in wide field, and as compared to that, the super resolved image where you see more structure, but where you also nicely see that background has been removed. Another modality that Jörg came up with is image scanning microscopy, commercially known as AiriScan. Zeiss implemented that in their product range. And that was yet another clever idea of Jörg. And basically, this is his first publication on the image scanning microscopy. And the idea is, again, pretty simple. This is the emission of a point-like emitter. What you do in confocal microscopy is you use a pinhole, you filter out background, and you typically put that setting of that pinhole at 1 ARRI unit. And you all know that confocal has a slightly better resolution than wide field imaging. And it turns out that by working in this condition, you absolutely not at the maximum of the resolution of confocal microscopy. If you would put the pinhole at, say, 0.2 ARRI units, you would have a much better resolution, but at the cost of a very poor signal-to-noise ratio. So what Jörg proposed is not to put a point detector, but an array detector, where each of the elements in the array correspond to 0.2 ARRI units, so that you also basically detect the fluorescence that you normally reject in confocal microscopy, and that you reassign the intensity of the neighboring pixels into the central pixels. And actually this works pretty good, and it's a relatively simple realization of super resolution microscopy. Finally, I want to briefly discuss with you on expansion microscopy, invented by Boyden at MIT, published, as far as I know, for the first time in this science publication in 2014. And note, this is the first time that the first publication was in one of the star journals. The other breakthroughs were all reported in what is considered to be less general and star-like journals. Anyway, if you want to see his explanation, I can recommend this TED talk. It's quite instructive. And basically what it boils down to is that he, instead of engineering the point spread function or come up with very complicated optics, he said we're going to reverse the problem, reverse engineer the problem. We're going to expand the sample, make it bigger, so that we can look at it in more detail. So the concept is pretty straightforward. It's also very popular in biology nowadays. Actually, I was a non-believer of this microscopy modality to start with, but I ventured into it with a few PhD students, and it's actually quite intensive line of research now, and I'll show you in a few slides where I think this can be of real help in biology. So what you do is you fix your cell, you label proteins or structures of interest with a dye. You also functionalize a polymerizable unit to the proteins or structures of interest in the cell. You add monomer, acrylamide monomer, you do a polymerization step, you digest all the structures in the cell, and you basically have created a hydrogel. So by adding water, it swells and it expands, typically a factor of 4. Key molecule to do this is this molecule over here with typical linker chemistry that we know from labeling. It's that way that you anchor the proteins of interest or the molecules of interest into the gel. It works. This is some work we did in the lab. You immediately see this is pre-expansion, post-expansion. Look at the difference in the scale bar. So it's a factor of roughly 3 to 4 that you expand. Very often you lose intensity because you basically dilute the labels. That is something that needs to be taken care of. And with better linker chemistry that I'll come back to in a minute, you see that you can really make nice images. You can do this on spheroids. Again, look at the difference in scale bar. And the linker chemistry I was referring to is we came up recently with what we call a trifunctional linker, so that we can specifically target more organelles or items of interest in the cell. So we are not limited to proteins or features that have free amine groups. And I think that is an advantage that was recently published. And I think we showed there for the first time that you also can go for expanding of membranes, and that opens up a few interesting research avenues. So to finish off, what's next? Well, I think you're going to see quite a bit of what's next in the rest of the program today. Look at the different things that are going to be discussed. Super resolution with micro lenses and so on and so forth. I think what still is needed is real-time 3D super resolution microscopy. There have been attempts in that direction. I think what Theo Lasse did a couple of years ago with SOFI, with this patented beam splitter cube. Was very elegant. So you get in real time super resolved images here. The color coding is depth of a filamentous network in a cell. And this is super resolved real time 3D. So that's interesting. New technical developments will definitely contribute to more widespread use of some of the techniques. There is a new implementation of MinFlux by the Tinnefeld group that will be published soon. I cannot show it because it's not published. I checked this morning. It's not out there, but it's coming, and that's really interesting. It's a pulsed interleaved implementation of MinFlux, really interesting. Combinations of technologies, we'll hear about that today as well. So, for example, this is Helge Ewer's work on coupling STED with expansion microscopy. Quite interesting paper from 2018. Like I mentioned, I think the expansion microscopy in conjunction with other super resolution modalities will prove to be very useful in what is called the field of single cell multi-omics, where you try to resolve in individual cells transcriptomics, proteomics, genomic information, epigenetic information. It's a very crowded environment. There's very many molecules. Multiplexing is key here. And the fact that you swell and clear the cell allows for this multiplexing approaches. So here's an example from my lab where we look at two MRRs in a cell with different copy numbers. And that works pretty well. And this is just 2, but you can do that easily 10 or 20 different messenger RNAs. And then finally, again, parallel with single molecule detection, single molecules really made it fast to the market. For example, the next generation sequencing of Pacific Biosciences, this big box here, is actually nothing else than an automated single molecule microscope. A little movie I will not play. Basically, it's single molecule detection in zero mode waveguides. And we hope that the same will happen for super resolution imaging. And actually, that is something we are working on. And research, I hope I can show you in somewhere in future. So we try to push for not really sequencing, optical mapping, a tool that is complementary to sequencing based on super resolved imaging. So super resolved optical mapping to target specifically the microbiome, which is still kind of a hard target for the regular sequencing approaches. So with this, I hope I have given you an overview of what is out there, what is still possible. So I thank you for listening to me. This was my perspective. Maybe I missed some things. I'm always happy to learn. If people say, you missed that, it's always good to learn something. So thank you for listening.